Okay, so can you guys can hear me, right? Figure this out. Okay.
Yes.
Yeah. Okay, great. So today we will be learning a module which is really interesting and it very connected to the next module, and this is on nucleic acids. And your nucleic acids can be DNA and RNA. And the next one is going to be on proteins, so linked. Okay, so what did we do last time? So I just wanted to start with, last time we were working on lipids. Okay, so the lipids are, um, there are different kinds and they can be divided into three categories based on, their differences in terms of storage, how they contribute in terms of structure or their function. So this is a really nice summary. It will help you recall what you did last time. And I have to say that lipids is sometimes gets very confusing. So nucleic acids after that is such a refreshing change. It's really fun doing it. And like I said, feel free to interrupt me. And if you don't understand something, and if you feel uncomfortable while I'm talking and you don't want to interrupt me, please put your question in the chat box. So we are here to learn about the important types of nucleic acids and what are their biological consequences when human beings are subjected to radiation. How does that impact nucleic acids? So we're going to start with their structure and definition because that's what we have been doing with all the other modules. So nucleic acids, they are large biomolecules. They're essential to all known forms of life. Everything that we know of has nucleic acid. Okay. So want to make sure that we understand how important this is. So their primary function is to store genetic information, and they're also responsible for passing on the information. So there lies the most important molecule, because you can have the body doing so many functions and use vitamins and proteins and different things for different purposes, but only one molecule can pass on the information. And I think you're all aware of that. So the monomer, what are the monomer or the monomeric unit? Single unit is nucleic acids are made up of nucleotides. And let's pause and take a look at this really nice visual on the right side. If you see nucleic acid structure, then one of these unit, let me just highlight it using this highlighter. One of this unit would be a nucleotide or a monomeric unit or a monomer. Okay. So this unit is responsible, it is the monomeric unit. It's responsible for making the entire polymer, because you can have different variations of that unit. And where is the variation? Do you see that top right side? And those variations are the ones that result in different types of of the monomer units. So don't have to worry about this phosphate group and P and O right now, because we're going to study those in details. Nucleotides are monomers that make up the nucleic acid, such as DNA or RNA, and they are connected to form nucleic acids. So with this, we're going to go to the second slide, where we are actually going to look at each of this monomeric unit. And I'm going to go ahead and delete that so you don't have to see that. Okay. Okay. So have you guys done the structure of DNA and RNA before?
Yeah, I think in biology we did.
Okay. Okay. So yeah. So you did in some of the subjects you might have studied this before. But let's try to look at closely at these definitions, and I'm pretty sure they're important also from your quiz and exam point of view. So what is a nucleotide? A nucleotide consists of three different parts. One is the base, which is also called the nitrogenous base. The second one is the pentose, which is the 5-ring sugar or the carbohydrate that you studied. And the last one is the phosphate. So these are 3 different parts. And if you see the number 1, that's the nucleotide. The entirety of all of these 3 things put together is a nucleotide. Now, what is a nucleoside? If you don't think about the phosphate, just this, we'll come to the word purine or pyrimidine. Don't worry about that. Just the base and the sugar, those 2 combined is the nucleoside. And if you just think of the base, that can also be called, though it's rarely called that, but nucleobase or nitrogenous base. So that is how these nucleotides and nucleosides are arranged. So there is a slight difference in their definitions, and you can see how they could be really important from a quiz standpoint. Now, if you see here, there is purines and pyrimidines. I'm sure there is more mention of those, so we're not going to worry about them right now. But is there anything, if there is anything that is not understood in this slide, or if you want me to repeat, like I said, you can interrupt me or write that in the chat. Okay, so now the next one, we are now going to take a closer look at what is the base, because these bases are purines or pyrimidines. Now, what are these molecules? Why are they important? So these are the ones that are forming the hydrogen bonds which keep the two strands connected. Okay, so that's one of the ways of getting the 2 strands connected. So these are nitrogen containing heteroaromatic molecules. So nitrogen, yes, we have it. Heteroatomic means there are not just one type, but different types. And aromatic is a ring. So that explains these 2 structures. They're almost on one plane. That is, they're not like, 3-dimensionally. They're more like a paper, like imagine a paper. So they're more like a planar structure. And they have another interesting property. They absorb UV light around 250 to 270, which is very useful in many experiments and diagnosis and everything. So for now, we have to remember that pyrimidine, which has the longer name, has only 1 ring, and purine, the shorter name, has 2 rings. So I think you all know that nucleic acids are linear polymers, because that's what we were discussing, especially looking at this one. So they are polymers, just like we did polymers in carbohydrates. I'm thinking about glycogen and starch, which are polymers of single unit of glucose. Just like that, nucleic acids are linear polymers of nucleotides. And there are 2 types. One is DNA and the other is RNA. So the DNA is the short form or abbreviated form of the word deoxyribonucleic acid. And it provides the entire code for the cell's activities. So just to give you an example of the extent, I'll share with you that each one of you has, trying to think, has about between 60 to 100 trillion cells. Okay. Now let us take one of the figures. Let us say you have 60 trillion cells. In the 60 trillion cells, each cell contains DNA. But how much DNA? So you know the DNA is in chromosomes, and you know some of those things. But if you put all of those DNA together, let us say you're able to tape them together, and you're going to put one after to get them, and make a linear strand out of all the DNA. And it's 6 feet of DNA packed in one cell. And like that, there are 60 trillion cells. That's a lot of DNA. So this enormous amount of DNA packs a lot of information and allows the cell to do its activity, and allows the organism in turn to do its activities. So the RNA here, or the ribonucleic acid, is important because once the DNA needs to get that information in its code out to proteins, the intermediate form is RNA, or the ribonucleic acid. and it converts the code into proteins to carry out the cellular functions, which means that DNA and RNA are one of the most important molecules in cell biology, and they're responsible for storage and reading of genetic information of all forms of life. So this really helps put a perspective that they are very important. And so all the more we need to make sure that we learn about them, as you know, you are going into those fields which is directly connected to preservation and of human life. So DNA and RNA. So DNA carries the genetic information in the cell, like we just studied. It contains instructions for body cells to perform specific functions, and the sequence of nucleotides determine the individual hereditary characteristics. So you know some of these things already. Now let's come to the part which is like where you are actually doing some thinking in here. So DNA is converted to RNA using the or by the method of transcription. And this transcription is the process that is like universally present in all organisms. After the DNA is converted to RNA, RNA is converted to protein. Okay, so if you see here transcription and translation, can you think of a way to remember that transcription comes first and then comes the translation? Can you think of a way to remember that? Is there any, like, do you have any secret trick to remember that?
Somewhere I read that just alphabetically transcription is before translation.
Anyone else? Sorry, I was trying to mute myself. Okay, so yeah, that's right. That's how I remember it. And the way I remember it is that it is transcription alphabetically comes first. So if you see here, transcription is TRNSC and this is L. So it is definitely, you know, you can arrange it alphabetically. And this is a popular quiz question. So I'm pretty sure that you're going to see this question somewhere or the other. Okay, so transcription converts DNA to translation converts RNA to proteins, and that's what I think is written out here. So I want to give you a minute for you. Okay, okay. So first comes transcription and then comes translation. Okay, good. So we have it here. So if you see here, there is a process here that conversion is done. So this pairing is T with A and A with T. Are you all familiar with this kind of pairing?
Yes.
Okay. I think this is going to come later, but that's fine. I'm going to still go back and just cover on this, that A pairs with T and G pairs with C. I'm just going to write here. That's Chargaff's rule. So and G pairs with C. Now, I would also say that this rule is valid, but for DNA, but what about RNA? Is it the same?
No.
Okay, what is the change in RNA?
The U, which is uracil, right?
Yeah, A pairs with U. Sorry, it was a. So in DNA, A pairs with T, but it can also pair with U when it comes to RNA. And it's also just, you know, RNA is a single stranded molecule, which means that, you know, you won't be able to see the double strand, but you will be able to see the U in there instead of T. So that's why if for any reason, if this is something that you have read a while ago, it's important for you to know that RNAs don't have any T's. Okay. So once this is accomplished, then comes the next part, that is, there is a 3 nucleotide codon that you make, and that codon can be converted to the amino acids, and this amino acids table is discussed later. Okay, so if you see here, DNA is a fundamental molecule found in living things. So we did some of this, but what we have not done so far is that DNA is actually having many different important parts. One of them is that there is a set of DNA sequence which is responsible for encoding for a gene, and that gene actually can transcribe, and then translate to a protein. Now, if you see a particular chromosome, there are many, many genes in there. But if the chromosome, if you see here, it is condensed with the help of histone protein. So you see those little beads, they are histones, which are positively charged proteins. With them, with the help of that, that is condensed. And that condensation is what leads to the formation of this famous chromosomal form, which you are very, very familiar with. The one thing more to remember is, I know that everyone is so familiar with this chromosome form where you have these like X-like form, but remember that this is not how the chromosomes are always present. Can you tell me when are chromosomes present in this condensed form? There is only one particular stage of cell when they are present like that in a condensed form. Do you know what stage is that?
Are you talking about like either in mitosis, I
Yes.
think, right?
That's exactly what I meant. Thank you, Lisa. So we're talking about mitosis is when the chromosomes are actually condensed. Otherwise, they are not really as present as condensed chromosome, but rather as chromatin, right? The chromatin material is loosely present in the nucleus, and it is actively transcribing, and that transcription results in eventual translation to proteins. So if the chromosomes are always tightly bound like that and they're condensed, then that they cannot really transcribe and make proteins. Okay. So this is an important from the standpoint as I was explaining to you. So imagine you have 60 trillion cells, of which each nucleus is just 0.05 millimeter. That's how small it is. And in that, these chromosomes, if you put all the DNA together, that's 6 feet of DNA in every cell. That's a lot of DNA. Now, most of the cells have them in diploid sets, at least when we talk of humans, it's diploid, but there are other organisms, there are plants which can be polyploid. For example, let's think about strawberry. Did you know strawberry has 8 sets of 1 chromosomes? Like, wow, 8. So have you guys ever done or seen people doing strawberry DNA extraction?
Yeah, I did it in plant lab before.
You did that before, yes. Okay, Lisa.
Yeah.
Anyone else? Like, I'm pretty sure this is something, it's a popular experiment. Like, a lot of people might have even seen it in some kind of a symposium or, in something like that. So, strawberry DNA extraction, very, very doable at home. If you have never done it, I highly recommend you doing it. You can easily find the protocol in the internet. And if you have any kid who is at home who is upwards of 4 years, they will really enjoy doing it too. So involve them and do it. And strawberry has 8 sets of of any chromosome that they're present is in copies of 8. So coming to the terminology, this is important again from the quiz standpoint. So I'll keep telling you what is important for you is haploid. Haploid means just 1 copy of chromosome. And diploid means 2 copies of chromosomes. And then there is polyploid, which is multiple copies. And those are shown by the figures in here. So there is a question for you on the bottom. So if the haploid number is 23, and then what is the diploid number, which is, oops, sorry. Did it stop sharing the screen? Can you guys?
Yeah, we can still say.
Okay. So if the diploid number of an organism is 50, what is the haploid number?
25.
Can you say that again? Sorry.
I think Tiffany did say the answer. If the diploid number in an organism is 50, what is the haploid number? The last question line on the bottom, I'm not sure if you're able to see the last line. Sometimes it gets a little tricky. Can you see it now?
Yeah.
If the diploid is 50, what is the haploid number?
It's just half, so it's 25.
25, yes. Okay, good. Let me bring this back on here. Okay, so yes, it is 25. And so please don't be thinking that haploid is half and diploid is double. Haploid is 1 and diploid is 2. So this terminology, we have to understand it well, because if we understand it well, then we never get anything wrong. Okay, now comes RNA. So the convenience with the DNA was that there is only one type of DNA. It's double-stranded. We saw that it has 4 bases. We're going to see a little more. But the RNA comes in 3 different forms. So there is messenger RNA, there is ribosomal RNA, and there is transfer RNA. Their pictures are shown below. So the messenger RNA is the one that actually describes the genetic code from DNA, which means that after transcription, what we get is the messenger RNA. It gets all the information from DNA, and then it exits the nucleus and comes to the cytoplasm. So if you see here, this is the messenger RNA, like just a prototype, just looking at the picture of messenger RNA. And then it is converted to protein with the help of ribosomes and transfer RNA. What is ribosome made up of? So have you heard of ribosome before? Like, what do you know about ribosome? Like, it's organelle, like mitochondria is a powerhouse. Like that, what is ribosomes used for?
I think that's where the protein is synthesized or
Great.
made.
The protein is synthesized? Yes, perfect. So the ribosome is made up of ribosomal RNA. And that is the second type of RNA. It helps the translation of messenger RNA into proteins. And it has 2 subunits. Depending on whether it's a eukaryote or a prokaryote, those subunits may be different. Then there is, finally, what happens is you need to get all those amino acids from the, from the cytoplasm. So transfer RNA is the one that acts as this molecule, like a messenger. It brings in all the amino acids and it helps in the translation. So the process of translation is not shown here, but 3 important components of translation are shown here. Messenger RNA, the one that converts to protein, ribosomal RNA, which helps make the machinery that converts the mRNA to protein, and transfer RNA, which brings in amino acid one by one to construct that protein. So all the 3 have some essential role. So if for any reason their roles are not clear or you would like me to repeat them, just put it in the chat and I'll be happy to do it for you. So now after this overview, we are going to take a closer look at the structure one more time, because now, I always believe that if we go back and forth on a concept, then it may help us to understand the concept better rather than putting all the details in the first few slides and then not talking about it till the end. So if you look here, the word deoxyribonucleotide, I'm sorry, that is a, oops. Sorry, looks like There is a typo here. I want to fix it. I don't want to make that mistake of having a typo. Okay. So deoxyribonucleotide, it is the name that is comprised of different parts. So let's try to understand first the deoxyribo or the deoxyribose part. Focusing only on the blue part. That's the sugar. What kind of sugar is this? This is ribose. And why it's called deoxy? There was another hydrogen, oxygen here, which is gone. So it is deoxygenated. And that's why it's deoxyribose sugar. And the phosphate group is right here. And the base is on the top right here. Now, my question to you is, out of all the three, which one of them gives a charge to the DNA? And what is that charge? You can also say there is no charge if you think there is no charge. Is there any charge in here that you can see? I can clearly see a charge.
Yeah, negative charge.
Okay, Lisa, and negative charge, and I'm pretty sure you're seeing on the phosphate group. Is everyone seeing this place where there is a little charge on the, let me show with the pointer. Do you see this negative charge in here?
Yes.
Okay, so you can imagine that when there is phosphate molecule or group in every DNA monomer or the monomeric unit, you can imagine that DNA has to be negatively charged because there is nothing else canceling that charge. Okay, so that is one important part of it. Then let's see the other one. Let's go to the next one. Okay, so when we talk about the bases, which we started talking about in the previous one, and let me see. Yeah, so these bases, what are these bases? So let us take a closer look. We're taking a closer look, and I'm going to tell you a trick to remember what is what. So there are 4 bases, which is adenine, guanine, cytosine, and thymine. And if you look here, the guanine and adenine are purines, and pyrimidines are cytosine and thymine. Look at this structure, look at this names carefully, pyrimidine, cytosine, thymine. Do you see all 3 have Y's in them? Do you see that pyrimidine has a Y, cytosine has a Y, and thymine has a Y? So,
Yeah.
yeah. So that's how I remember. Whenever I think of pyrimidine and someone says, hey, which ones are pyrimidines? And I'm like, the ones with the Y's in them. Okay. So you can remember by that cytosine and thymine. And since uracil does not have a Y, I agree with that. But it is so closely related to thymine because it just replaces thymine. So uracil also falls in the pyrimidines category. Whereas the purines are the one, though even though the smaller name, but double, there are 2 rings, aromatic rings, and which makes this a different kind of structure. Now, remember this as we go on to the ribonucleotide, which is not deoxy, just the ribonucleotide RNA. Okay. So if you take a look here, so we were just talking about the structure of DNA, and we said that DNA has deoxyribose sugar. So you see that OH and H. We are going to compare this with RNA now. You can see that this has OH and OH, which means there are 2 intact hydroxy groups. So which means that the building block of RNA is a different kind of sugar, even though the rest of it is same. It's a ribonucleotide. You know, this is something I've always wondered. Why is everything made up of DNA and not made up of RNA? Can you think of a reason what it could be? think of a reason. I think I have read that RNA, even with that very tiny change, just that one hydroxy group, becomes a little bit unstable. Okay. And that instability does not make it a suitable molecule for the purpose of inheritance and for passing it on. Whereas that single change makes DNA highly stable. It's very surprising how much of one hydroxy group can make that change. That's why chemistry is so important and interesting. And so if you know, I mean, I know people who work with, I mean, if you work in a pathology lab, of course, you're going to store DNA at 4 degrees or minus 20, but DNA is perfectly stable at room temperature. DNA is stable even if you heat it to a good extent. So naturally, it survived all these millions of years, rather billions of years, in different organisms. Okay, so coming to now the nucleotide bases. So I'm going to draw parallels with the DNA. So if the DNA has purine pyrimidine, that is, the purines are the guanine and adenine, and here the pyrimidines are cytosine and thymine. Here, the only difference is the uracil, like I said. Okay, so it's nitrogenous base, and that's the uracil is the only difference. Okay. So this is a good point to pause, especially since 2 more people joined a little later. I want to just reflect on this and say, we are talking about DNA and RNA and how their structures are. And one thing that is really important for DNA and RNA is the basis. And there is purines and pyrimidines are the one that form the basis. So the way to remember that, everyone, is that pyrimidine has a Y and the cytosine and thymine has a Y in it. Okay. So this Y is how we remember that pyrimidine are part, I mean, cytosine and thymine, and the uracil is also part of pyrimidine because it is similar to thymine. This is an important from your quiz or exam standpoint. I really want you to know this. So I'm going to ask you again, and I hope every one of you can score well in this part. So here we go. I think this learning trick should be now really, really easy for you. What is adenine? Is it a purine or pyrimidine? It's uracil is purine or pyrimidine. Which one of them is what? So to summarize, the building blocks of DNA are A, T, G, and C. Two pyrimidines and 2 purines. And RNA in contrast is A, U, G, and C. Okay, now to answer the question, what are the components of ribonucleotide, you might have to go back and take a look, but if you remember it, it's very simple. It's the sugar, it's the phosphate, and it's the base. Okay, so with this, I think this is easy. Okay, so DNA versus RNA. Now we are in a great position to do a comparison. It's also a good point for review and for anyone who wants to ask any questions or just say, I joined late, I don't understand this. Feel free to interrupt me, okay? DNA is double-stranded. I really like that part. DNA, double-stranded kind of go together. So it's very easy to remember. RNA is single-stranded. DNA has a deoxyribose as the sugar, whereas RNA has the ribose as the sugar, which is the differences between the OH and the H right here. The bases that are used are let's start from the bottom, G, A, C, and then T for DNA. And for uracil is G, A, C, and U. So these are the 3 common bases, and this is one different base. So this is a really important slide. So if you have any questions, you can ask me, or you can just put it on the chat. I think this is clear. Discovery of DNA is a really, really fun and interesting, almost like a novel by itself. I'm not sure if you're familiar with this. And if you have time to watch this, but HHMI Bio Interactive is a wonderful resource. I'll try to see if I can put some videos for you. I think I have some videos put in there. Biointeractive has a whole movie on discovery of DNA. It's a 17-minute video movie, and it kind of recaptures how the DNA was created, I think. So I encourage you to watch this if you can. Also a very fun thing to watch with kids who are interested in science, like a little bit slightly older. So did you know that there were even speculations that the DNA was a triple helix? There are all sorts of speculations being made before the structure word. So let's look at this picture on the right side. Rosalind Franklin and Morris Wilkins, they were supposed to work together for the X-ray diffraction part. They were supposed to help solve the structure of DNA. And, you know, Rosalind Franklin was one of the few women scientists working in the field. She was, you know, often probably intimidated, maybe. And so she had a very not so friendly approach to the other male scientists she was working with. Watson and Crick, on the other hand, were working to find out the structure of DNA. And one day, when Rosalind Franklin was not in her lab, but she did have a structure, the X-ray, you know, picture of the DNA in her drawer, then Morris Wilkins showed Watson and Crick the picture of Franklin's X-ray diffraction data. And immediately Watson and Crick realized, because, you know, one of them had this kind of background that he knew exactly what they were looking for. So immediately they knew that it was a double helix. So the discovery can be credited to all the four scientists, Watson, Crick, Morris Wilkins, and Rosalind Franklin. Unfortunately, just before the Nobel Prize was to be awarded, Rosalind Franklin died and she never got recognized that way. But the structure was out in 1953, and it was really, really important because it led to, you know, a new generation or a new era of in biology. So DNA is a double helix that stores genetic information. That is the conclusion they came at. It was also that Erwin Chargaff had put some rules together. Those were extremely helpful. If Chargaff had not put those rules together, also, you know, Watson and Crick would not have been able to solve the structure. So that DNA-based composition is between species. And what do we mean by that? We mean by that some species have lots of ATs and others have lots of GCs. DNA from tissue of the same organism have the same base composition. DNA-based composition in organism does not change with age or with environment or nutritional status. In all DNA, in all species, A pairs with T and G pairs with C, so which means amount of A is same as T, amount of C is same as G, which means that amount of purines is same as pyrimidines. Okay, so I'm going to ask you something here. We're talking so much about DNA, and I told you like every cell of your body contains the same exact DNA, same genome, which you inherit from your parents. Why is your cells of your eye different from cells of your muscle? Why? Can you think of a reason? You can come up with any random reason. You didn't have to be right or something. You just have to, I'm just encouraging you to think. Can you think? I mean, this question might have come across your mind, right? So the question here is, why is your eye cell different from nerve cell or from your, I don't know, muscles and your skin cells? Why are these different?
Function?
Functions are different. Good point. But functions are decided by what? I mean, what is the basis of those differences? Is DNA different in eye cell and muscle cell? You can take a guess. You don't have to be right. You just have to take a guess.
No. Are
So
they?
the DNA is same. DNA is same throughout your body. Every single cell of your body, all the 60 trillion cells have the exact same genomic DNA which you inherited from your parents. Exact same DNA. Why are these cells different? What is that particular molecule that makes it different? Is it okay? I'm going to give you a hint since you guys are so quiet. And like I said, you can just say something, and even if it's wrong, it doesn't matter. Like I don't care if it's wrong. All I care is you try. So is it DNA? Is it RNA? Or is it protein?
DNA.
Protein.
Protein.
The RNA.
Okay. Let me put it like that. So good thinking. I got all the three answers. So let me put it this way. Different genes are expressed in different cells. So the amount of DNA and the type of DNA is same. What changes? What changes is the amount, is the genes that are getting expressed, which means that the type of RNA that's going to be made will be different, and definitely the type of protein is different. That is why your eye cell is so different from your muscle cell, all being decided by finally by the protein. So Lisa, you were right when you said functions, and the only thing I have to say here is those functions are governed by the proteins. But I think Nancy said RNA, and that isn't wrong either, because RNA is the reflection of what genes are being transcribed. So what I want you to do is appreciate, it's a complex thing going inside the human body, which we haven't really, which you might not yourself know what's going in your body. So that is the proteins making all the difference. Okay, that was a good pause. Let's come back to it. According to Chargaff's rule, base pairing takes place between purines and pyrimidines. We talked about it. I want you to appreciate that AT has 2 base pairs. Look at this, hydrogen bonds, 2 of them, whereas GC has 3 hydrogen bonds, if you see here. Okay. So this is an example of how Chargaff's rule plays. You can see A pairs with T, T pairs with A, C pairs with G, G pairs with C. You can see that.
I have a question about the pairing. So are you just demonstrating it in your slide, or do they like A and T at the top and then it's T and A? Like, is that on purpose, or like, do they always reverse? You know what I mean?
No, they don't have to reverse.
Okay.
And a good question, Olivia, because if you see here, you only read one side of the DNA. So you are reading only the 5' to 3', which we are not going into details, but you read only one strand of DNA. So actually, let me show you when you're reading it.
Oh, so the codons you read up and down. Oh, so.
I wish I had a better slide, but I can still show you. Yes, so I'm going to block off one side so that you're not confused. And sorry about the red color, didn't mean for it to be red. Sometimes it's like so helpful to lie. Okay, so it will be reading as A, T, C, G. That's it. That's how this DNA sequence will read. And you will say, what about the opposite one? Well, we can make it, A pairs with T, T pairs with A, this pairs with G, and this with C. That's it. And you have gotten, I'm trying to use the eraser, and you got your thing back. So you're only reading one strand of the DNA. And you can appreciate here that I did not remove the deoxyribose sugar and the phosphate. So you can see that. And why is it double stranded? 2 reasons. You can see clearly 2 reasons. I'll circle those reasons. Reason 1, this hydrogen bonds right here in the middle, guys, this is the one responsible. And this phosphate bonds on the sides. 2 reasons for double stranded. Hydrogen bonds in the middle, hold it in the middle. Phosphate bonds on the side, hold it on the sides. That's why the DNA is double stranded. RNA, unfortunately, doesn't have any of these. So nothing like this forms. Okay, so this is just showing the same base pairing, just for you to appreciate how it goes in a longer strand and what are the kind of distances. This is angstrom, which means it's 10 to the power minus 10 meters. It's like really, really small, only observable by X-ray diffraction pattern. And that's what they use for the discovery of the structure. So these are called complementary strands. Complementary because we know A pairs with T and T pairs with A and G pairs with C. And yes, they didn't quite do it purposely, but you're reading one side, so then it will come like that. And in here, I think I made the life easy for you. And take a look at this very, very carefully. I'm trying to block this off for your convenience. Look on the top. It starts with C-A-A-T. That's what is here, C-A-A-T. C-G-T-C-A, C-G-T-C-A. So I'm trying to show you, you read 5' to 3', and this is how you write it down. So if someone shows you just one strand of DNA, don't be confused. Don't be confused like it's supposed to be double. Why didn't they show double? You can immediately create the complementary strand. No one needs to tell you that. You can just do the pairing, Chargaff's rule, and make AT and GC pair and form those strands. Also worth appreciating here is that they're running anti-parallel, which means that it's a little bit harder to explain. So look at the direction of the sugar in here. So the direction of the sugar in here is the sugar is looking down. Let me see, show that. Okay, so if you look at do you see how the sugar is looking down here? And in here, the sugar is looking up. You know, so just for you to appreciate, it's anti-parallel. So if you have the hands, like both hands together, but in the opposite direction, and that's what is the anti-parallel structure. So they differ a little bit in structure, and this is based on the numbering. And I made these colors match so you can appreciate how, they are. I want to pause here and ask, like, if you have any questions for this thing. Okay. Okay, so what is a gene then? We talked about genes and we said different genes are expressed in different cells. So what do I mean by that? So basis of the information, one single unit is called the gene. It's called the unit of heredity. Like what is every unit of information you you are inheriting, is from a gene. So human beings have 3.1 billion base pairs of DNA, and they have about 30,000 genes. How much percentage of DNA is useful in making this gene? So you do have lots of DNA, but only 1 to 2% of the DNA that you have is used for making genes. So you might ask me, what about the rest of the 98%? What does it do in human body? Nothing. We don't know. Maybe it does a lot of things. We don't know. Right now, because we don't know, not because it's less important or something, but there are a lot of duplications. There is a lot of extra DNA. We call it junk DNA. And scientists are trying to determine what does it actually do. So out of the 100% DNA you have, 98% does nothing. Only 2% is useful. Now comes the part which is important for you to understand from your quiz and exam point of view. So let's pay a little bit attention. What is a codon? Codon indicates a sequence of 3 nucleotides that code for only one amino acid. So how does that happen? After DNA is converted to RNA, which is what is shown here, then RNA goes out of the nucleus. And this conversion is something you can explain by the periodic table. Look at the first sequence, UGG. Go to the first base, U on the red, G in the blue, and G here, UGG, tryptophan, which is indicated as TRP here. Let's go to the next one, UUU. UUU is a phenylalanine, which can be indicated as PHE. I hope you can see that there is a correlation between the 3 nucleotide codons and the amino acids. My question is, do you see the correlation, or is it something that you want me to explain to you? Okay. You're not expected to remember the table. You can, you should be just provided with it always, like forever. No one expects anyone to remember this table. So there are three ways of writing the amino acids. You can write the full name, phenylalanine. You can write 3-digit one, saying PHE, or 3-letter code. You can write single one, which is F. And why is phenylalanine F, even though it starts with the P? Because you see the P is already taken by proline. Okay, I'm going to show you here. It's taken by proline. Okay, so that is why some of them, even though leucine, and there is lysine, I'm trying to find. Oh, this is the leucine, and here there is lysine. Lysine is K because leucine has already taken the L. So some of them have those variations. Okay. So this is for you to try out. This exercise is for you to try out. I can do it if you want me to do it. So I want a little bit of your response here, like yes or no. Should I complete it? And or is this something you are thinking like, wow, this is like so easy. Why waste time on this? Or you can even write on the chat like, yes, do it.
Yeah, can you do an example?
Okay. So look at this. I have done for you a little bit, but I'm going to explain it. T becomes A. A becomes A, I mean, not becomes, I'm sorry, T pairs with A or complements A. A complements U. Remember, there cannot be any T in RNA. C with G, G with C, A with U, and T with A. Now, we have to go ahead and you have to help me here. Okay, so someone help me. T complements with what?
A.
A, and then next is also an A. Then what about G?
C.
Okay, so now we have found a AAC. So we are going to look for this AAC in here. So follow me as I do this. A, and then go here A. So A and A should be here. I found the AAC. It's asparagine. Okay. So again, no one expects you to know this. So asparagine is N. You just write the N. Okay. So what we have done is we have kind of taken care of this part. Okay, so now I'm just going to change the color and go to next one. CCT. What becomes of CCT? C pairs with, so remember the rule, A pairs with, oops, A pairs with U pairs with A, C pairs with G for anyone who came late, or G pairs with C. That's the rule. So C pairs here with, what should we do here?
G.
So we can do G here. Oops. Okay, G, and then G, and then the last one is a, what should we do with T? There is, okay,
A.
pairs with A. Sorry, I should have done that. Now, so this is our new one. Let's look for it. G is on the bottom, G is here, and GGA right here. Okay, G. So do you see how I'm doing this? Does anyone have any questions how we are proceeding ahead with it? C pairs with the, so make it a different color. C pairs with G, T pairs with A, and G pairs with C. So next one is GAC. It will make me take like forever because it's just the process is so slow. GAC, find it here. That's the aspartic acid. So that will be D. But do you see how I'm slowly converting the DNA to RNA and RNA to protein?
Yes.
Okay, good. Thank you, Nancy. So, which means that, I mean, it's not that hard. I'm trying to see if I can see. I don't know if I could save it or not, but it looks like I can clear off the drawings now. Right now. Okay. So this is what I want you to make sure that you understand this, and it is in one of your learning checks. Convert DNA to RNA and RNA to protein. So my request to you, please try it out. Okay. So you can see very easy question. What is transcription and translation? What are the differences? Convert the DNA sequence to RNA and protein for this particular sequence. Looks like there is a small mistake here. Shouldn't have been you. I'll update this PowerPoint and put it there back there. I know some of you have already read it. Okay, so nucleotides and nucleic acids, what are their functions? Nucleotides, the functions, those individual units, their functions are, they're used as energy for metabolism, which is, we know ATP is the energy currency of the cell. They can serve as cofactors for various enzymes. They can also be used for signal transduction. That's an error in here. Then as far as their functions go of the nucleic acids like DNA and RNA, they are responsible. So these are single units. But when you look at the actual polymeric units, there's storage of genetic information, transmission and processing, and protein synthesis. So this kind of sums up some of the basic things that we were learning. Now we are left with more easier part. So we can go a little bit faster in here. Can DNA get damaged? Yes, it can. It does get damaged all the time. Oxidative damages inside our human body result in 10,000 mutations per cell per day. Wow, that's a big number. And it gets repaired because of the DNA repair machinery, the proteins in DNA repair that help, start a pathway in which the mutated DNA can be repaired back to original form. And when it is unrepaired, either the cell dies, which is the senescence, or the apoptosis, but it can also lead to progression of cancer. So many things can cause mutation. You don't even need an exposure. Things that are happening, oxidation is happening inside your body there when the food is getting metabolized. And it's not metabolized, catabolized, and it is forming all the smaller molecules. There can be replication error. There can be so many reasons. So what is a mutation? It is changing of one, it's removing of the right nucleotide to the wrong. And you may say, what do you even mean by that? Suppose A is supposed to pair with T. What if by mistake, A is paired to C? Oh, that could cause a change in RNA. Change in RNA could cause a change in protein. Change in protein can actually trigger even certain conditions. For example, sickle cell is a result of one nucleotide mutation. And sometimes the mutation doesn't do anything, and sometimes it does a lot. Okay? So we have to be careful that mutation doesn't always mean it leads to something fatal, but it can lead to certain conditions. Okay, so there is UV light, there is ionizing radiations. They can cause the rings to open up or strands to break. And they're not very easy to fix. And they can lead to, like I said, they can lead to different conditions. So here is an example of what can happen, formation of pyrimidine dimers induced by UV light. And that's why they say, like, you shouldn't be out in the sun for too long, or it can result in those kind of dimerizations. These dimerizations can result in, when unrepaired, can result in skin cancer. Okay, so those are kind of problems that can happen. And then when there is a kink like that, the structure of DNA is a little bit compromised. If you can see here, it's not original as this form on the bottom looks like. Then there is ionizing radiation, which can directly damage the DNA, or it can split a water molecule, which can cause damage. So direct, indirect, both kinds of action or mutations can happen. And how does this work? There is the radiation therapy work. We just learned that radiation is bad for the DNA, but the radiation therapy works because they damage the cancer cells. They kill the cancer cells, and that results in, them getting eliminated. However, it does destroy some of the good cells too. So, but when there is a mutation in the normal cells, they can repair themselves. Now, when we are thinking about applications in your fields, which you are going to go, is patients are treated with radiation therapy and in different kinds of conditions. They are also, it's also used as an effective tool for many, many years. And after 2/3 of cancer patients receive radiation therapy, they also, they may also have conditions in which the normal cells can undergo mutations and result in, certain other cancers. But, if it is treated effectively and caught early, then the radiation therapy can be very effective. Okay, it is done by, that it's done by oncologists who are trained to use this. Okay. So now, as far as people who are going to allied health, especially the nursing side, this is more relevant. There are different kinds of radiotherapies. There is external beam or internal radiation. There is also the more popular and getting more popular, I wouldn't say more popular, is the proton beam therapy, which is very localized. And as a result of it being so localized, it doesn't really damage a lot of cells which are in the neighboring. And as a result of which, it is much more, it's getting much more popular now. So this is something that we discussed in some of the other slides, the different uses of radiotherapy to cure, to after resection to prevent the reoccurrence, before surgery, palliative. There are different ways of which it can help. And these are the different instruments or the techniques by which, these conditions can be, it can be detected. Diseases can be detected. Finally, this is something that applies to both people in allied health and you guys going to the dental hygiene field, that there are x-rays are always needed, and there are different types of x-rays that which will be recommended based on whatever the condition is. You can read about them a little bit, and if you want, I can provide you with more articles, or you are going to read about them more as you go to your fields. There is also CT scans and fluoroscopy, which can be used, which are different types of procedures. They can be also used for the same purposes. Okay, I'm going to exit this. Let's see if I can stop sharing. Okay, so I'm going to pause the recording now so that we are at the end of the module.